Excellent. Good morning, friends. Good morning, good morning, good morning. So you should have had access to the fundamental data structures notes, these notes. And there were two lectures, two movies that went through these points. The ideas discussed in these lectures were a few fold. They were a list of experiments often performed in biotechnology. The premise, the idea was to understand these techniques. So the lectures that you watched discussed multiple experiments, okay? And there are many more. There are many different types of experiments. And as graduate students, you guys need to start learning how to combine experiments to answer scientific questions, okay? As graduate students, you guys need to learn to combine experiments to answer scientific questions. In English, we call this a experimental pipeline, okay? A pipeline is like a pipe through which liquid flows. This is called a pipeline, And you have to learn how to do experiments in a pipeline. Like maybe you do PCR, then you do agarose gel, and then you do Sanger, okay? And then maybe after that, you do an SDS page with the goal of answering questions, okay? You need to learn how to do this. So I want to give you an example. I want to discuss an example experimental pipeline. Okay, so let's give a common experimental question. A common thing that you might deal with is detect the presence or absence of an organism, okay? Or simply detect an organism. This is a very, very, very common experimental question in science and in biotechnology, okay? So an example of this would be detect a pathogen, which causes sickness, or detect a symbiont, or detect a parasite, okay? These are common experimental questions, and I'm going to show you an experimental pipeline that you can use to answer these questions. So let me use a very, very specific example from my own research. Okay, so this is an example. So in chickens, in poultry, which is chicken production, there are parasites, and these parasites are nematodes, okay? They look like this. The species is Heterachus gallinarum, okay? These are parasites of chickens. And if farmers have parasites in their chickens, they lose money, okay? So if you have parasites, you lose money because the chickens lay fewer eggs. And if you have a house full of chickens, okay, the parasite can move from chicken to chicken. And one hypothesis is that there are beetles which carry the worms. So inside the beetles are the nematodes, okay? And when the chicken eats the beetle, it goes into the chicken, it gets in the gut, and then the worms get into the gut of the chicken. Okay, so hypothesis. Beetles spread chicken parasites. That's a hypothesis. So do you see how the question we want to answer? The question we want to ask is, do beetles carry the parasite? Do beetles carry the parasite? Do you see how this is an example of detect the presence or absence of an organism? Okay, detect a pathogen, detect a parasite. This is an example of that. Do beetles carry the parasite? Okay, so now I want to talk about a pipeline to answer that question. Extract DNA. Okay, so typically you collect the beetles into a tube, collect the beetles, smash the beetles. We put glass beads in the tube, and you shake the tube, and you shake the tube, and the glass beads will smash the beetle, which yields some stuff. Okay, and then step 3 is an organic extraction. Usually for DNA, it involves phenol, chloroform, and ethanol precipitation. So that yields DNA. Okay. Step 2. Once you have the DNA, you can do a PCR, which we discussed in the last class. Okay. But you need to design PCR primers. What do you want the primers to recognize? If the goal is to detect the parasite in beetle DNA, what should the PCR primers recognize? What should the PCR primers recognize? Write it in the chat. Another way to say it would be, what should the PCR primers bind? Write it in the chat. Okay, a target gene sequence. So somebody wrote target gene sequence. Okay, but which gene from what organism? From what organism? Okay, remember our goal. Our goal is to detect a parasite in beetle DNA. Because the question is, do these beetles, do they carry these nematodes? That's the question we're trying to answer. Yes, parasites. It should recognize the parasite. Okay, so if our goal is to determine, do these beetles carry these worms? We can answer this question with PCR of beetle DNA with primers that bind the parasite, the worm. Okay, so here's the worm. In the worm is a genome of DNA. Let's say we design PCR primers to recognize a sequence in this nematode parasite DNA. Let's say the distance between these primers is 200 base pairs, okay? So when we do a PCR, the quote unquote amplicon, the amplicon size will be 200 base pairs long, okay? So 1, extract DNA. 2, run a PCR on, actually I should say, with parasite primers. What's going to be step 3? Write it in the chat. What will be step 3? How about this person? This person. Are you there? What's step 3? What is step 3? Yes, good. Very good. Agarose gel. Okay. See, do you see how what I'm trying to teach you is how to string together experiments, this, etc., to answer questions. Okay. So step 3 is going to be an agarose gel. Okay. So we will run an agarose gel. This will be our negative control. This will be our positive control. And let's say this is sample 1 and sample 2 and sample 3. Okay. And we will run a ladder, and let's call this 50 base pairs, 100 base pairs, 200 base pairs, 300 base pairs, 400 base pairs, 5, 6, 7, etc. Negative control should have nothing. Positive control should have a band at 200 because the amplicon size is 200 base pairs. Okay. Let's say the results look like this. My question is, which sample has the parasite? Okay. Sample 1, sample 2, or sample 3? Which sample has the parasite? Very good. 2. Very good. Sample 2 has the parasite because its band matches the positive control and it's right at 200 base pairs, which is the size of our amplicon. Okay, so we conclude that sample 2 has the parasite. And because sample 2 is DNA from a beetle, we conclude that beetles carry parasites. Okay, so if we want to answer the question, do beetles carry parasites? Is the answer yes or no? Let's hang on. Let me, if the gel looks like this, do beetles carry parasites? Yes or no? If the gel looks like that. Very good. No. If the gel looks like this, now, do beetles carry parasites? Yes or no? Yes. Good. Good. You guys understand. Very good. Okay. So do you see how we are answering scientific questions? We are answering scientific questions with a series of experiments. Okay. But let me ask another question. How can we be sure that this band is from the parasite? Okay, so this band, how can we be sure that this band is from the parasite? What's going to be our fourth experiment? Okay, so if our experimental pipeline is 1, extract DNA, 2, PCR, 3, agarose gel. Thank you, Piahu. 4, what's going to be our fourth experiment? How can we be sure that this band is from the parasite? Sequencing. We need to sequence it, okay? This band on this agarose gel just tells us size and it tells us presence, but it does not tell us sequence. We can't be sure. We can't be sure that the band is from the parasite unless we sequence. So the fourth step is sequencing. What kind of sequencing? Which sequencing should we use? Which kind of sequencing? Very good. Sanger. Because remember how I wrote Sanger is used to sequence one thing, one amplicon. Okay. When we look at our gel, there is one amplicon. So if we want to sequence that one amplicon, we will use Sanger. Okay, very good. So let's walk through how Sanger works. How does Sanger sequencing work? Let's talk about how Sanger sequencing works. Okay, let me zoom out and get this. Okay, let's talk about this. So this here is a deoxynucleotide triphosphate, a DNTP. We discussed this in the first lecture, okay? This is a special, let me spell this correctly. This is a special NTP. It's different because at this position, there is only a hydrogen. So there's no oxygen. So let me ask a question. This one is called a dideoxy. A DD NTP, dideoxynucleotide triphosphate. So let me ask a question. What will happen if I add a DD NTP to a DNA molecule instead of a DNTP? Yes, very good. Yan Lu, +1 point. It will stop extending. Why? Let's draw it out. So remember how DNA is linked together. Okay. Remember the carbons. This is carbon 3, and this is carbon 5. Okay. DNA is linked at these carbons. Okay. And this oxygen is required to bind the phosphate of the next base. Okay. So if you add a DD NTP to the end of a DNA molecule, it cannot extend. Okay. It cannot extend. Now, this is very, very important for understanding Sanger sequencing. So how does Sanger sequencing work? Okay, so let me explain how Sanger sequencing works. You take a tube, and in the tube, you put DNTPs, a lot of DNTPs, and a few DD NTPs. Okay, these go into this tube. So ingredient 1, ingredient 2, 3, you also have a template. So let's add our parasite amplicon. That's about 200 base pairs. Let's also add 1 primer, okay? And they will add a polymerase into this tube for Sanger sequencing. Okay, so here's our template. This is what I call template. This is our 200 base pair amplicon. Okay. Then in Sanger sequencing, they will do a PCR cycle. Okay. So they will denature to anneal. Let's say here's our primer, 1 primer, 3 extend. Okay. Now let's think about the extension when there's a lot of DNTPs and a few DDNTPs. Okay, so the DNTPs will be black and the DDNTPs will be green. Okay, when the polymerase starts writing, most of the bases will be DNTPs. Okay, but at some point, 1 DD NTP will get added. And after that, it cannot extend. Okay. And this is a random probability to add a DD NTP. Okay. This is important. There's a random probability to add a DD NTP. So this PCR reaction will produce, the result of this will be some PCR amplicons that are short, and some will be longer, and some will be longer, and longer, and longer, and most will be middle size, and some will be very, very, very long. And each of these will end with a DD NTP. And these are all different sizes, okay? So this will produce different size amplicons, okay? And in Sanger, oftentimes the DD NTPs will shine colors. They'll have fluorophores. So let's say a DD NTP A will shine yellow, and a DD NTP T will shine orange, and a DD NTP G will shine red, and a DD NTP C will shine blue. Okay, so each of these will shine a different color at the end, depending on the base. Okay. Now, let me ask a question. How can we separate these DNA molecules based on size? How can we separate these DNA molecules based on size? Write it in the chat. Very good. Agarose gel. Okay. So in early Sanger, in classical Sanger, they would take these and they would run a very, very, very long gel. And you would get bands of different sizes that would shine different colors. Okay. And from this, we could know blue was C, 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 C. Yellow was A, 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 A. And red was G, 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 G. And do you see now this? This is our sequence of DNA. So when you do Sanger, here's a question. When you do Sanger sequencing, what do you get back? What does the data look like? What does the data look like? Okay, you get two different types of things. One, you get a .txt file, which is the A, T, G, C, etc. Okay, to N. And this text file comes directly from this process. Okay, so you get a text file with the sequence. Second thing you get, what's called a chromatogram. Okay, a chromatogram looks like this. Chromatogram will look like this, something like this. And you can learn a few things from the chromatogram. The y-axis is the fluorescent intensity, the peak intensity. Okay, so a very, very, very high peak is good data. As you sequence longer and longer and longer and longer reads, the peak intensity will degrade. Okay, so long, we call this long reads. This is like greater than like 500 base pairs. Sequence will degrade in quality. Okay, this is bad data. This is good data. And this will work about 500 base pairs. Okay. So let me ask a question. What if we saw this? What if we saw blue equals G, A, C, and let's do yellow. And this will be T. So my question is, what is the sequence from this chromatogram? Okay, spend 2 minutes, figure out the sequence from this chromatogram. What is the sequence? Take 2 minutes, figure out what is the sequence. Okay, this is kind of a trick. This is kind of a trick. In English, we call this a trick question. Trick question. It means that in English, that means the answer is complicated. Okay, so we would think that, let's see. So here there's a T, here there's a G and a G. Here there is a and an A. But what about this? What about this spot? There's actually both. At that spot, there is both C and there is also T in the sample. What does this mean? Okay, what does this mean biologically? What could this mean? What do you think? You're thinking correctly. You're on the right track, but the answer is not a mismatch. Mutation is closer. Mutation is close. Yeah. So yes, very good. Okay, Piao Hu. Very good. Heterozygote. Very good. Good. Okay. The answer is it's a polymorphism. We call this in English a polymorphism. Or you might have two quote unquote alleles. Okay. So imagine there's a gene X that codes for blue eyes. And imagine if you have a mutation, you can get brown eyes. And because humans are diploid, we have two copies, okay? If you did a PCR and a Sanger sequencing reaction studying this gene, and the human had both copies, let's say got this one from mom and this one from dad, okay, you would see something like this. You would see a polymorphism. It means it's a heterozygote. A heterozygote. Let's talk a little bit more on this example. Let me show you some specific data. Okay. So here, this is data checking beetles for parasite DNA. Let me actually move this. I'm going to move this. So these are real data. These are chicken farms. These are chicken farms. Okay. And we collected beetles, and we ran a PCR. So we collected beetles from this one, A, this one, B, this one, C, this one, D. Okay. So here's A, live beetles, A, dead beetles. So these ones come from this barn. B, live beetles, B, dead beetles. These ones come from this one, etc. And we have two PCRs. Two PCRs, okay? Nematode, which is the parasite. And we have a PCR of the insect, which is the beetle DNA. Okay. So we would conclude that this beetle has the parasite. This beetle has the parasite. In B, none of the beetles had parasites. There were none. So B was no parasite. In C, this beetle had the parasite. This beetle had the parasite, etc., 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 etc. What about D? Does D have the nematode parasite? Write it in the chat. Yes or no? Very good. No. Okay. So A has it and C has it. So A is infected and C is infected. Okay. Let me ask another question. What is the purpose of the insect PCR? These primers recognize beetle DNA. What is the purpose? What is the purpose of the insect PCR? So that's this one, 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 and this one. What is the purpose of this insect PCR? Why did we do that? Write it in the chat. Yes, it is it's a control. What does it tell us? It's a control for what? A control for what? Let me look at this band here. Here. In this lane, there is no insect DNA band. What does that tell us? What does that tell us about that sample? What does that tell us about that sample? If there's no insect DNA band, what does that tell us about that sample? Write it in the chat. Yes, very good. Very good. Very good. It tells us the DNA extraction was bad in that sample. Okay, it's a control for DNA extraction. Okay, so if there is no parasite band, but also no insect band, we can't conclude anything because the data is bad. The DNA extraction was bad. Okay, so it's only in samples where we have a good insect band that we can conclude anything. We can conclude that this beetle was negative. This beetle is not infected. Okay, and this beetle is infected. Okay, but this one here, this is just bad data. Bad data. Okay, so this is very, very common. You will see this when you see PCR. When you see PCR data, okay, you will always see one gel, which is the test, and the test might look something like this. And you will always see a control for DNA quality, which should look like this. Okay, so PCR data, you always need two experiments. You need a control for DNA quality, and then you need the actual test. Okay, very important. Controls are very important. Okay, everybody have a great evening or day. You guys, it's your day. Have a great day today. I really appreciate you guys, and I'm having so much fun teaching this class. You guys are great students. So thank you for participating. Have a good day.
I shall meet death head on. Decade, you've got to get out of here. Beware foul demons and beasts. There are many foes here. The time has come to cleanse my horde. This